Hello. So this is for the University of Dallas MicroBiology course with Dr. Cody. This is for the disease report assignment. I will be doing the Zaire Ebola virus. So some basic taxonomy and information. The Ebola virus is part of the family Filoviridae of the genus Ortho-Ebolavirus. The subspecies and common name is the Zaire Ebola virus, part of group 5 of the Baltimore classification scheme. So it is a negative single-stranded RNA virus. It's characterized by hemorrhagic fever or the inability to clot one's blood. Up here we have a diagram showing the genomes of viruses within Filoviridae. The first 5 are part of the Ebola virus genus. And what you'll notice is that the overall genomes of viruses within this family have the same gene sequence, these rectangles, as well as the same open reading frame, and so which are your arrows, but they differ in the number and position of gene overlappings, so these triangles, as well as intergenic regions, and so there are differences between these, but they are generally the same in a lot of senses. Down here we have a bit of a phylogenetic tree displaying viruses within the family, specifically these different genuses. Ebola virus is right here. There are 6 known to man, 6 different strains, 5 of which can affect humans. The one I will be focusing specifically on is the Zaire Ebola virus, also known as EBOV. So characteristics of the infectious agent. So Zaire Ebola virus is classified as a membrane-enveloped filamentous non-segmented negative single-stranded RNA virus, which is a bit of a mouthful. It contains a nucleocapsid with a diameter of 80 nanometers, and a virion with the envelope has an overall size ranging from 800 nanometers to 14,000 nanometers due to the filamentous nature of the virus. So there's quite a wide variety in the size of this virus. Viral entry is mediated via macropinocytosis, which is a form of endocytosis similar to phagocytosis, in that the glycoprotein will bind to the cell surface, and viral particles will enter into what is called a macropinosome, which is kind of like a vesicle, which enters into the infected cell, breaks apart in the cytosol, releasing viral particles for replication. So just some virulence factors. There are several different virulence factors that the Ebola virus will actually undergo. One of the main ones is your VP40 complex, which mediates that macropinocytosis mechanism. So VP40 is a critical matrix protein, which in addition to regulating viral transcription, coordinates virion assembly and budding from infected cells, does several different other things. Mechanistically, VP40 interacts with the plasma membrane of the host cell, driving formation of new virions, and interacts with the cell factors to facilitate the budding process. So here, this diagram just shows the dimer form of this VP40 complex. And over here, we can see a diagram showing the glycoprotein or spike protein of the Zaire Ebola virus. Take note in these specific regions, like the glycan cap of the glycoprotein, which will be targets for specific treatments and vaccines. So reservoirs for infection, the WHO believes, it's not for certain, that the original and host carrier of the virus are fruit bats of the family Pteroptidae, who will then secrete or excrete waste full of nuts, seeds, fruits, things like that, which other animals such as chimpanzees, gorillas, and gazelles will eat. And then we as humans will eat these animals or drink infected water, thus infecting ourselves. So the virus is originally zoonotic, and then once humans become infected, it's transferred via bodily fluids, contact with infected or contaminated objects, as well as from parent to child, specifically within the womb. A lot of this can be due to deforestation and basically encroachment on the habitats of these animals. Meaning that these animals come in closer and closer proximity to humans, resulting in a higher frequency of outbreaks. So pathology and the immune response. So the virus typically enters the body through mucous membranes or skin ruptures, initially infecting the dendritic and macrophage cells of the immune system. The virus then replicates and spreads to your regional lymph nodes and then spreads further through the body through viremia, which is basically the virus in the bloodstream. Through viremia, infection continues to a variety of cells, including your endothelial, your hepatocytes, and many immune cells. Take note that endothelial cells line the organ cavities of many different parts of your body, which means there's a high chance of infection to several different organ systems. Initial immune response is triggered by toll-like receptors, or TLRs. Various mechanisms, however, including the VP35 and VP24, or viral protein, inhibit the production of interferons, a critical signaling molecule in the immune system, thus weakening the immune response. So the body just cannot see the virus, and this subversion results in prolific replication and further infection. So symptoms, so incubation of the Ebola virus is typically between 2 and 21 days, on average 8 to 10 days. And the Zaire Ebola virus victims exhibit a myriad of symptoms. So early stage, you have fever, muscle and joint pain, nausea, vomiting, diarrhea, and general abdominal pain. Hemorrhagic manifestations include mucosal hemorrhaging, gingival bleeding from the gums, gastrointestinal hematemesis, and hematoptysis, or coughing up with blood. Neurological defects result in are things like headaches, disorientation, seizures, and rarely encephalitis, which is inflammation of the brain. And then your advanced symptoms include hypovolemic shock, coagulopathy, multiple organ failure, and a cytokine storm resulting in systemic inflammatory response syndrome, which is basically when the body, understands that it's highly infected, and it throws everything the immune system has at the virus. This is very detrimental to the host, and very often results in death. So diagnosis of the virus is quite difficult, because many of the symptoms that are shown are are seen in other viruses and other diseases as well. So the initial diagnosis questions revolve around patient history, specifically whether this patient has been in contact with a person who has or have had the Ebola virus, or has this patient been in the geographic locations wherein Ebola virus carriers are prevalent. After that, there are several lab tests which can be done, such as the polymerase chain reaction, or PCR, which is one of the primary methods for diagnosing Ebola virus infections. So PCR can test for genetic markers of the virus and are highly sensitive and specific, making this a credible method. You also have antigen capture, enzyme-linked immunosorbent assays, or the ELISA assays, which test for antigens in the blood of potentially infected individuals. And it is used in the early stages of infection. You also have things like serology assays, which test for antibodies to the Ebola virus in the bloodstream and are generally used in the later stages of infection because antibodies take time to develop. So over here we have a diagram showing your PCR, and over here we have the ELISA assay, which tests for antigens versus specific genetic markers. So treatment and vaccines. So these are quite recent, as the treatments and vaccines for Ebola virus were set and approved in around from 2016 to 2020. So they are quite recent. Your main treatment method approved by the FDA is Inmazeb, and it's composed of a mixture of 3 monoclonal antibodies, and so the Zaire Ebola virus uses its glycoprotein to mediate cell surface attachment, subsequent entrance into the host cell, and each of the antibodies in Inmazeb targets a different non-overlapping epitope on the glycoprotein, thus preventing cell surface attachment, further replication. It also prevents the inhibition of immune responses. And so the immune system is able to actually see and recognize the virus as something harmful and react accordingly. The vaccine, which is also known as the RVSV-ZBOV, has been approved by the FDA as a vaccine for Zaire Ebola virus victims, approved for individuals of 12 months of age and older. The CDC states it is a replication-competent, live, attenuated recombinant vesicular stromatocys virus, which basically means that the virus is able to self-replicate. It contains live version of the virus, such that the immune system will be able to present itself. However, it's weakened in that it contains only one gene from the actual virus, specifically the gene that encodes for the glycoprotein. And the vaccine utilizes the RVSV virus, a virus primarily affecting animals as its base, which has been modified via recombination processes. And this is generally resulted in positive results within clinical trials. Clinical efficacy has shown rapid antibody response within 14 days after a single dose, and the clinical efficacy was verified during the 2014 to 2016 outbreak within Guinea. So pertinent outbreaks, the Zaire Ebola virus has been around since 1976 when we first uncovered it. We've had outbreaks all the way up to 2022. However, there's only been one main outbreak. The other outbreaks have resulted in total cases reported less than roughly 300. And this is because these outbreaks generally happen in rural, isolated areas within Africa, where transmission is difficult to get to large populations of people. However, in 2014 and a little bit at the end of 2013, there was a major outbreak in Guinea, which then spread to neighboring countries, Sierra Leone and Liberia, transforming it into an epidemic. This was mainly due to poor public health infrastructure within these countries, as well as very strained healthcare organizations, the inability to efficiently survey these outbreaks and control them. So within this time frame of 2014 to 2016, you had basically international partners like the WHO and the CDC who sent aid in multiple different forms to Guinea, Sierra Leone, and Liberia. These organizations also helped forge new relationships between these organizations and their governments, such that they can actively look for these viruses, for these outbreaks, and prevent them. Fortunately, the vaccine and the treatment methods have largely helped stop potential outbreaks from happening. However, you have to remember that in these countries, they're still developing, and so access to this infrastructure is really limited. This specific outbreak tallied a total of roughly 25,000 total cases reported, with a total of 11,000 plus deaths. However, this number is most likely lowballing, just because the surveillance was just not nearly as good as it should or could have been. And so here you just a graph of the three different countries, Sierra Leone, Guinea, and Liberia. And this is for cases reported. And so you can see we pass, that 25,000 mark eventually as it goes up. So future outlook and prevention. So fortunately, with the development of approved treatment methods and vaccines, the risk of pandemics or subsequent epidemics and subsequent pandemics are greatly minimized. However, you have to take into consideration that within these countries in Africa, the poor socioeconomic factors are a great contributor toward this virus spreading, as well as physical environments, health behaviors, and healthcare, which is very limited in these developing countries. So access to the vaccines and treatment methods has to be improved even now. In addition, things like climate change and the human deforestation and taking over of these natural habitats of these animals greatly impacts the frequency of these outbreaks, because these animals are just coming closer to us as we encroach on them. And so things like this still have to be prevented. It should be noted that in general, the risk of pandemic for this virus, for Ebola viruses in general, is very low because of the methods that we've developed against them. However, we still need to put further efforts into helping these countries in Africa, these third world developing countries that just don't have the infrastructure, the money, the workforce and all these different factors to really contain these viruses.